If you're like me and you're looking at an animal population, you might look at it and say, I wonder what alleles these particular animals have. I wonder what genes are in these animals. I wonder if these are heterozygous or homozygous recessive. So in this population of frogs, you might wonder, what are the exact alleles? Well, what we're going to do today is going to help you on your journey of discovering the particular alleles and genetic makeup in certain populations. So in the video that we're going to watch, I'm going to go over the basics. What's a gene pool? What's inside a gene pool? I'm also going to take a look at how we calculate frequencies of different alleles. Then we're going to take a look at Hardy-Weinberg theory and how we can actually numerically calculate evolution and predict what's happening in a population. And then we'll wind up this video solving some actual Hardy-Weinberg problems. So let's get started. So let's start by talking about populations and gene pools. The first definition I want to give you is population. You may remember a population is just a group of individuals in a certain location that can interbreed. So in this example here, I've just got a little graphic of a population of humans. And you may also note that I've given you the genotype of a particular trait. Let's say it's eye color or hair color. Eye color and hair color are actually quite complicated, but we'll just go with that for an example. So this individual here is heterozygous, right? They've got one dominant, one recessive allele. This one has two recessive, so they're homozygous recessive, and you can see the rest. So if we were to take, and we wanted to know what's the gene pool of this population for hair color or eye color, we could take all those alleles and put them together in a group. Or one might say a pool of all the genes together to see collectively how many dominants, how many recessives are in this pool of genes. We call that, of course, a gene pool. It's just the collection of alleles in a certain population. Now, maybe you're saying, Herder, remind me, I don't remember, what's an allele? So you may recall that humans are diploid, right? We have two sets of DNA. We have one that we inherited from mom, one that we inherited from dad. So on that DNA, you've got certain genes, and maybe from mom, you inherited a dominant version of that gene, maybe a dominant version of a hair color. And from dad, maybe you inherited a recessive. So an allele, remember, is just an alternate version of a particular gene. So one might want to figure out, in a gene pool, what is the frequency of the dominant allele? How frequent do we see the recessive? So we can calculate allele frequency by just seeing how common that allele is in a particular population. And we can get very fancy with our diagrams, and we can say, okay, for this particular gene here, it started at the frequency, all these genes started, they were at about 0.5, so 50%. But over time, over several generations, maybe one gene went down, one frequency, and then the frequency of another arose. You can see in this example, it's not quite as sporadic. Some go down a little bit, some frequency of genes go up. What you're looking at here is just evolution. You're looking at changes in the frequency of alleles over time. So, if we want to calculate some frequencies, we have to first start with some definitions. So in genetics, we use the letter P to indicate the frequency of the dominant allele. We use the letter Q to indicate the frequency of the recessive allele. So take, for example, a certain population of frogs where brown is dominant to green. So in this case, you see all the brown frogs, you see a few green. So if brown is dominant, green is recessive. So notice, to be a recessive frog, you have to have both recessive genes. Now, in this case, notice, to be brown, you could be homozygous dominant, two big B's, or you could also be heterozygous, one big B, one little b. How would we go about calculating the frequency of the dominant and recessive allele? Well, all we got to do is count them and divide by how many total alleles there are. So let's start with the big B's. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9. I'm counting 9 of the dominant B's. Let's see here about the little b's. I got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. I count 11 of the little b's. So to be able now to calculate the frequency, there's 20 alleles total. So the dominant allele, also known as P, is equal to 9 over 20. The recessive is equal to Q, which would be 11 over 20. And I just happen to have a handy-dandy calculator, so let's just calculate that. 9 divided by 20 is 0.45. So our frequency for the dominant allele is 0.45. That means the rest have to be recessive. So let's do that. 11 divided by 20, 0.55. We can see that the frequency of dominant is 0.45, the frequency of recessive is 0.55. Now, I'm going to give you a little hint about something in the future. If you add those both up, it should be 100%. And we're going to look at an equation that will tell us that in a minute. So, when we're trying to calculate evolution, when we're trying to calculate these frequencies, there is a theory that we use. It's called the Hardy-Weinberg theory or Hardy-Weinberg Equilibrium, named after two scientists. Actually, one was a mathematician, one was a scientist. And they came up with this theory of a hypothetical non-evolving population. So imagine a population that was at equilibrium. It was not evolving. We can use that as a model to measure evolutionary change over time. So for Hardy-Weinberg equilibrium to apply, for this hypothetical situation to apply, there are several conditions for a non-evolving population. These have to be met. Now, all of these have to be met. Of course, they rarely ever are, if ever, but for us to do this math, we have to assume that we have a non-evolving population. So the first thing that has to happen is we have to have a very large population size, meaning no genetic drift, which of course means there couldn't have been a bottleneck, there couldn't have been a specific event to create a founder effect. None of that could have happened because we need a large population size. We also can't have any migration, no movement in or out, right? This needs to be a stable community. No migrating across the globe. There can also be no net mutation, so we can't have a lot of genetic changes because really what we're looking for is a stable population for this hypothetical Hardy-Weinberg equilibrium to take place. So the next condition that has to be met is that there needs to be random mating, meaning there can be no sexual selection. So we can't have peahens going after the peacock with the most feathers. No sexual selection can be taking place. And then finally, as Darwin would tell us, there can be no natural selection in this example, right? Darwin, of course, was all about natural selection. But in a Hardy-Weinberg equilibrium example, no natural selection can be taking place. So these are all the rules. These are all of the conditions that must be present for us to be able to do these math problems so that we can measure evolutionary change. Now, as I mentioned, these are seldom all present, right? So this is a hypothetical example to help us model some evolutionary change. Okay, so in Hardy-Weinberg equilibrium, we're going to assume for our first examples that there are two alleles, a dominant one and a recessive one. Okay, in this example, I'm using B's. You can use whatever letter you want. The frequency, of course, as you remember, of the dominant allele is P. And then we talked about the frequency of the recessive allele is going to be Q. And because the total frequencies must always add up to 100%, like we just saw with the frogs, we can come up with a formula that says the frequency of the dominant allele plus the frequency of the recessive allele has got to be equal to 1 or 100%. So let's do an example. Some little herd fish. I call them herd fish. Just made that up. And if you look at these fish, you might notice, okay, Some are blue, some are orange. Let's look at the trait for color. In herd fish, blue is dominant to orange. So if we're looking at these, we can sort of tell we got some blue ones here, we got some orange ones. If orange is recessive, that must mean that the genotype for this must be homozygous recessive. So if we're using B's, this would be little b, little b. There it is. For the dominant, they're both blue. Do we know what the genotype must be for them? Well, it can actually be one of two things, right? It can be homozygous dominant, or it can be heterozygous. So it's important that we're starting to look at these examples so that we can understand how to work these Hardy-Weinberg problems. So let's go on and try to do one. Let's look at some individuals now. The frequency of homozygous dominant individuals is p squared. So if you think about it, in terms of individuals, now we're talking about their genotype. In the last example, we were talking about alleles. What's the frequency of an allele? In this example, we're talking about what's the frequency of a certain genotype. In this case, homozygous dominant. That's going to be represented by p squared. The frequency of homozygous recessive is equal to q squared, because if you think about it, homozygous dominant has a big B and a big B, or two of the dominant. Recessive has the little b, little b, little b. So to get that, we're getting q squared. Since big B is equal to p in our example, little b is equal to q, that's how we get those numbers. And then heterozygotes is 2pq. You may say, I don't understand that 2pq. Why is there 2? Well, let me give you an example. We will come up with this formula then to account for all 100% of the genotypes. Because if you think about it, p squared is homozygous dominant, q squared is homozygous recessive, 2pq is heterozygotes. Well, these are our only options if we're looking at a completely dominant gene. So all those together must equal 1. This gives us our second Hardy-Weinberg equation. You may remember the first one was p plus q plus equals 1. We just looked at that. Here's our second one. So we've got two equations. How do we derive that equation? Well, it's actually, if you think about it, if you take two heterozygotes, and here they're using A's, and you take A over here, here's A for a heterozygote, here's the A from dad, right? There's the sperm, there's the egg. If you do a Punnett square cross, you'll see there's one that's homozygous dominant, one that's homozygous recessive, and two that are heterozygous. And since we know heterozygous is truly just a P and a Q, one dominant, one recessive, then you got to add these two together. 1, 2 of those gives us our 2pq. So if we're looking now back at the herd fish, and we're trying to figure this out, okay, if you're homozygous dominant, in this equation, what are you corresponding to? Well, homozygous dominant is p squared, all right? So we can do our p squared. Heterozygous frequency, the frequency of heterozygotes, is equal to 2pq, right there. And then, of course, our homozygous recessive is just going to be equal to q squared. So this is going to help us solve these problems. And I gave you the actual letters there to see as well. Okay, let's solve a problem. So here's a typical Hardy-Weinberg equation problem. Let's say we have a population of 100 herd fish. 84 are blue, 16 are orange. We want to know how many of each genotype. All right, so let's remember our two equations. We've got p plus q equals 1, p squared plus 2pq plus q squared equals 1. Remember, this is the equation for the alleles. This is the equation for the genotypes, or the individuals. So to solve this equation, we've got to figure some things out. And I'm going to give you a big hint. Always start, not always, but most of the time, most of the time, start with homozygous recessive. Because the orange we know are homozygous recessive. The blue, that's 84, that's these two together, right? But we know orange, 16, are all homozygous recessive. So we can start with those. So 16 out of 84, if you do the math, 16 divided, actually, that's not right. There's 100 total, so it's 16 out of 100 is the frequency of this genotype. So we can say little b is equal to 16 out of 100, which is actually, if you remember back, Q squared is our number of homozygous recessive. So this is equal to our number of homozygous recessive, which is also equal to Q squared. So if you do some math, that means 0.16 is equal to Q squared. So how do I find Q? Well, take the square root of both sides. So 0.4 is equal to Q. So our frequency of the recessive allele, is equal to 0.4. Now you can do some easy math. If 0.4 is the frequency of the recessive allele, or Q, I can take this down here and say P plus 0.4 is equal to 1. So that means P must be equal to 0.6. Easy enough, right? So now I know what my P is. Now I know what my Q is, and I can easily solve the rest of these. So let's just, I'm going to write them up here. We know that P is equal to 0.6. We calculated Q to be equal to 0.4. So to get the genotypes, to get the genotype of homozygous dominant right there, what we would do is we need to get P squared. So 0.6 squared would be 0.36, right? So 36% are this. To get the heterozygotes, we know that's 2PQ. So we're going to take 2 times 0.6 times 0.4. So 2 times 0.6, that looks like it's 0.48. So 48%, or 0.48, is the frequency of those. And we already figured out that the frequency of these was 0.16. So how many of each genotype? Because I was nice and gave you out of 100, there are 16 of those, 48 of those, and 36 of those. Okay? So the hint, always start with the homozygous recessive if you're given an example like this, because that's easy to solve. Okay? All right. So I'm going to let you try to solve this problem. So look at the problem, pause the video, work it out, and then I'm going to help you with it. All right? So hit the pause button. Okay, did you get it figured out? Let's do it. So in herding sheep, sheep that you herd, white fur is dominant to brown. So we know then, and they told you to use the W, brown will be little w. White, remember these are going to be big W, but they can also be heterozygous. So they could be homozygous dominant for white or heterozygous for white. They want to know what's the frequency of the dominant and recessive allele. Notice they didn't ask for the genotype, they asked for the allele. So we're looking at P plus Q equals 1. We're also going to use this formula P squared plus 2PQ plus Q squared equals 1. All right, so where was my hint? Always start with this one, right? So we know that 9 out of 100 is equal to the homozygous recessive genotype, right? Because 9 out of 100, that's their genotype. So that is actually, once again, Q squared. Q squared is the homozygous recessive genotype. So let's do some math. So if Q squared is equal to 9 out of 100, Q squared is equal to 0.09, which means, take square root of both sides, Q is equal to 0.3. So that is the Q is the recessive allele. Well, we're asking for the dominant and recessive allele. So if P plus Q equals 1, we can say P plus 0.3 equals 1. So P must equal 0.7. So what's the frequency of the dominant allele? 0.7. What's the frequency of the recessive allele? 0.3. Problem solved. Okay, one more problem for you. Do this practice problem, try it on your own, and then unpause me, and I'll help you work it out. All right, how did it go? Did you get this one? Same sort of deal. This one, though, we're looking at the percentage of the population or individuals that are heterozygous. You may remember heterozygous is 2pq. So we're going to use our two formulas once again. P plus Q equals 1. P squared plus 2pq plus Q squared equals 1. Remember my trick. Start with the recessive. So in this example, 25 out of 100 are recessive, right? So this is the homozygous recessive genotype. So 25 out of 100 is 0.25. So we can see, since Q squared is the number of homozygous recessive genotype, we can say Q squared is equal to 0.25, which means Q is equal to 0.5. So we can now take this back up to solve for P. P plus Q equals 1, so P plus 0.5 equals 1. That means P equals 0.5 as well. So the recessive allele frequency is 0.5. The dominant allele frequency is 0.5. But in this example, what we're wanting is the frequency of the heterozygotes. So what we've got to do is we've got to remember back to our genotype equation. 2pq is the heterozygote frequency. So let's come up here. So the frequency of heterozygotes is equal to 2pq, which is equal to 2 times 0.5 times 0.5. So to me, that looks like 0.5 or 50%. So 50% of these sheep would be the heterozygous phenotype. Okay? So hopefully this helped you solve some of these problems. The trick, remember, to solving these Hardy-Weinberg equations is really, I think there's three. First one, you got to remember P plus Q, that formula is about the alleles, and this formula here is about the genotypes or the individuals. The second big trick is really that you have to remember that in a case where you are given the number of individuals, you generally want to start with the homozygous recessive genotype, which is Q squared. Okay? So if you like this video, if it helped you on your journey of academic growth, then like and subscribe to our channel so I can continue to make some more podcasts for you.